Hey guys, welcome to Chapter 8. AP Biology, we're going to be talking about DNA this chapter. I'm going to move through this chapter reasonably quickly because, one, I think in general biology, you guys learned quite a bit about DNA already, and two, because this chapter is very, very brief. I mean, it might be 8 or 9 pages in your book, maybe less, so I would highly recommend you read it. It's a interesting read as far as textbooks go. There's a lot of experimental stuff in there, and it's just a real short chapter, so DNA. Let's jump right into it here. So, the intro story, like your book always has some sort of intro story, is about this hero dog, and the search dog's name's Tracker, spelt very cool, Tracker up here, located the last living survivors from the 9/11 attack on the World Trade Center. And that dog died of a degenerative neurological disease, probably due to the toxic smoke and the other things he breathed in at Ground Zero. And so his owner, this James Symington, applied for this grant sort of competition about the world's most deserving, animal who should get cloned, and he won that competition. And so they went on and they took some of Tracker's cells, and they made a bunch of clones that were identical to Tracker, and I think they had 5 puppies that ended up coming out identical to Tracker. And they inserted his gene into donor eggs, and the eggs into a surrogate mother dog, and then they had a litter of identical Trackers. So that was kind of a cool little introduction to the world of DNA. So for cloning, it's kind of a controversial thing, and it's something we'll talk more about in class. But very few of our clone mammal embryos will actually result in a live birth. A lot of the clones that survive have some serious health problems. We're starting to minimize this now, but basically, cloning animals is not by any means perfected. And basically, our problem is that DNA in our adult cells is controlled very differently than the DNA in embryonic cells. Of course, an embryo is this first little dividing cell after a sperm and egg unite to make a zygote. It becomes an embryo as it divides and starts to develop. And when you take DNA from an adult, let's say dog named Tracker, who's already however many years old, you're taking DNA that's already, 5, 7, however many years old, and then trying to put that into a new embryo. And so that can become a very difficult thing. And so as we continue to kind of perfect these methods, we get to the possibility of considering cloning humans. Is this technically possible, and ethically should we do this? And so that's a tough topic. We'll talk about it more. So let's talk about DNA, how we know where it is and what it does. The first guy to find DNA didn't give us a whole lot about it, but it was a long time ago, really, 1869. Johannes Miescher, he found DNA, and really all he did was he found that there was a chemical inside of the nucleus of a cell. He found that chemical was in the nucleus of other cells. And so he essentially said there is a chemical, and it's in the nucleus of cells. And that was about it. But it did lay the groundwork for some really cool experiments. So in the early 1900s, we get the Griffith experiment. This is a very famous experiment. And he was looking at pneumonia in mice. And in the 1900s, he transferred this hereditary material between dead and living cells. And so he had 2 strains of a pneumonia-causing bacterium. He had an R strain and an S strain. And so when he injected mice with pneumonia of the R strain, they survived. When he injected mice with the S strain, the mouse got pneumonia and died. And so then what he did was he took these S cells that he knew caused pneumonia, and he heated them up, boiled them, so that it would kill the bacterium. And he injected the mouse with the dead S bacterium, and the mouse survived. It didn't get pneumonia. And so then the really cool part. He takes the heat-killed S cells, which do not cause the pneumonia, and he mixed in the R cells that were alive that did not cause the pneumonia. And what they end up getting is the mouse gets pneumonia and dies. And he had live S cells. And so what this showed Griffith was there is information in this S cell, and if I boil it and I kill that cell, the bacterium R that wasn't causing pneumonia can somehow get that information from S, and R can become virulent. And once it becomes virulent, we call him S cells. And so he learned that there is something in this cell that is information that can be transmitted and can make a non-lethal bacterium lethal. And so there's his experiment. Very, very cool. The healthy R cells, no pneumonia. The healthy S cells, pneumonia and death. The heat-killed S cells, the mouse is fine. But if you mix the heat-killed S cells, which doesn't kill the mouse, with the R cells, which don't kill the mouse, you end up getting a dead mouse. And that was really the fascinating result, because we had figured out that something in this guy made it into this guy and killed this guy. So that's a kind of a cool thing to see. So, some other studies that led to our understanding of DNA. In 1940, a couple of scientists by the names of Avery and McCarty, they separated these deadly S cells from Griffith's experiments, and they separated them into three parts: lipids, the proteins, and the nucleic acid components. And they got this really cool idea that basically, let's destroy these one at a time. Let's destroy all the lipids and put it in the mouse and see what happens. And let's destroy all the proteins, put it in the mouse, and see what happens. And so what they found out was you can destroy all the lipids, and that heat-killed cell will still make the R cell virulent. And you can kill all the proteins, and those cells will still transfer information, and that mouse will still die. You can kill all the RNA, and that cell will still transfer information, and that mouse will still die. So by process of elimination, they found out the only thing left that we haven't destroyed is DNA. So the only thing that's moving from R cells to S cells, the only transmission of information, must be from DNA. It's the only thing we haven't destroyed. So the conclusion was that this transforming principle, this thing that changed a cell into something else, must be DNA. So let's look at some other studies. In the 1950s, we had a couple of scientists by the names of Hershey and Chase, and these guys looked at bacteriophage. Now, a bacteriophage is a virus, right? Bacterio, bacterium, phage, eating. So the bacterium eater, right? A virus that eats bacteria. And of course, that's silly because viruses are much smaller than bacteria, but he kills bacteria. And so they did this really cool thing where they were trying to figure out what does the virus put into the bacteria. And so they took the virus and they labeled all the protein parts with an isotope that they could track, sulfur 35. And they thought that stayed outside of the bacterium. Now the DNA in that virus, they labeled with a different isotope, which would show up as a different color, phosphorus 32. And they found that entered the bacteria. And so the conclusion was that DNA, not the protein, is the material that stores hereditary information. Because you have to bear in mind, the scientists of the early 1900s, they thought what you guys think and what I've taught you to think. If you don't know, guess protein, because proteins do like everything in cells. And so they thought when it comes to information and transmitting information, it's probably protein. Hereditary information is probably protein. So this idea that this very tiny, almost inconsequential DNA molecule was probably not the heavy hitter of information. It most certainly had to be some sort of protein. And Hershey and Chase showed that was not the case. That protein stayed outside of the bacterium, and that DNA went into the bacterium and killed it. And so here is your classic moonlander T4 bacteriophage. Gorgeous structure, right? Very cool head. Has like a ridiculous number of sides, like 20 or something like that. And his super cool little landing stations. And then this hollow sheath, which is this just horrifying little picture of like a needle. In goes the needle, the DNA up in here, shoots down through this hollow tube into the bacterium, and that thing is infected. Very cool. Happy for electron microscopy so we can see this cool little guy. So, We've got sulfur 35 isotope glowing red. He's outside of the bacterium. He shoots the DNA into the bacterium. The sulfur 35 is glowing outside of the bacterium. Bacterium sick. He didn't get the protein. Next trial, phosphorus 32. It's glowing red inside because it's now hooked to the DNA. The virus infects the bacterium. The inside of the bacterium is now glowing red. The virus is no longer glowing red. The bacterium gets sick and dies. The proteins leave, and because it's glowing red inside, we know that the DNA is what is transferred inside, not the protein. Very cool experiment. So there's the Hershey-Chase experiment. All right, so let's talk about DNA structure. You probably learned in Gen Bio that the building blocks of proteins are amino acids, and the building blocks of, fats are glycerols and fatty acid tails, and now we're learning that the building blocks of nucleic acids are nucleotides. And so we're going to talk about nucleotides. Nucleotide is the monomer unit for a nucleic acid. It could be DNA, deoxyribose nucleic acid, or RNA, ribose nucleic acid. Nucleic acid should consist of a 5-carbon sugar, 3 phosphate groups, and 1 of our 4 nitrogenous containing bases. So DNA consists of 4 of our nucleotide blocks, 2 pyrimidines called thymine and cytosine, and 2 purines, adenine and guanine. A bonds to T, C bonds to G. I remember this by South Dakota's economy is almost purely ag. Adenine, guanine, pure. We're purely agriculturally based here. That's not necessarily true, but it helps my brain. If it helps you, go for it. If not, get rid of it. Your call. Purines and pyrimidines. And so here you have adenine and thymine, and you can see why we've grouped our purines and our pyrimidines. Our purines, ag, have a double carbon ring. Maybe I shouldn't call it carbon. Nitrogenous base ring. There's a lot of nitrogen in this. Double ring for our purines, a single ring for our pyrimidines. Other than that, they're very similar. All 4 of them, have 3 phosphate groups. Lots of energy there. It might remind you of ATP, triphosphate, triphosphate, triphosphate. Adenosine triphosphate was ATP, and now here we have deoxyadenosine triphosphate. So very similar to ATP. We have our sugar. That's this orange piece here in all 4 of these. And then we have the nitrogenous base. And so our nitrogen-containing base, or our nitrogenous base, that's what really gives the identity to the nucleotide. What makes adenine adenine and guanine guanine is their nitrogenous bases. You see they're different. Guanine has the double bonded O in the amine group. Adenine has the amine group, and that's it. So they're different. So here's some cool rules. This is called Chargaff's rules. And they're very, very logical when you think about it. Chargaff found that the amounts of thymine and adenine in DNA are the same, and the amounts of cytosine and guanine are the same. So that's rule number 1, which makes sense because, like I just told you guys, A bonds to T and C bonds to G. And so as much adenine as I have must equal the amount of thymine I have because they bond together. Guanine amounts must equal cytosine amounts because they bond together. And what this allows us to do is, with very little information, we can actually calculate this. So let's say I told you I had a DNA sample, and that DNA sample was 20% adenine. I asked you to solve for all of the other remaining percentages. And you'd say, well, that's not very much information. But you know that adenine bonds to thymine. So if there's 20% adenine, there must be 20% thymine. So right away you'd have 20% thymine. Now, 20 and 20 makes 40. We've got 60% left to make up, and they have to exist in equal proportions between these 2 nucleotides. And so you'd say, okay, then if I have 60% remaining, 30% must be guanine, 30% must be cytosine, and you would have solved for all the proportions of the nucleotides, and you would be correct. So the next thing he found, this is Chargaff's second rule, is the proportion of adenine and guanine differs among species. So you and I don't have the exact same amount of A and G as a deer or a cow or a lizard or a fish. Ours is close to the same amount of adenine and guanine found in other humans. So perhaps that part's not quite as surprising as the first, but there you have it. So now here's where the DNA story gets interesting, and I've got a really cool video I'll find for you guys that kind of tells this story. But the discovery of DNA's actual structure that you see in your textbook, this whole double helix twisted ladder business, that was actually kind of a tough road for these folks to figure out. Franklin, Watson, and Crick are all involved in this, and here's kind of how pieces of it unfolded. So Rosalind Franklin is a very, very gifted scientist who worked with X-ray crystallography, which was basically firing beams of X-rays through a hardened crystal, and based on the diffraction patterns, she could get an idea of what sort of shapes the molecules were in. And she found these pictures of DNA. I shouldn't say she found. She took pictures of crystallized DNA that gave this banded X pattern that was signature of a helix. And so she had a photograph that showed that DNA was very likely a helical molecule. And this was the final piece of information that Watson and Crick needed to build their own model of DNA. And they had been trying to build a model for quite some time, and we didn't know how DNA was actually structured. We didn't have electron microscopes at the time, so they went back to just honestly building models, building big models out of wire and cardboard, and people thought the bases must be on the outside of the molecule, because why would they be on the inside? Enzymes couldn't read them, and so a lot of this was theoretical. And once they got Rosalind Franklin's photograph, they were able to put their model together very quickly. And so Watson and Crick are in your book, and in all science books for biology, because Watson and Crick published first. And that's a cold, hard reality of science. Everyone knows Charles Darwin, but almost nobody knows Alfred Wallace. Same idea. Darwin published first. He gets the credit. So I guess you can chew on that idea for a while. So Watson and Crick DNA model says that a DNA molecule consists of two nucleotide chains. They run in opposite directions, and they're coiled into a double helix. Their base pairs form on the inside of the helix, and they're held together by hydrogen bonds. Hydrogen bonds are a nice thing for DNA because they're pretty strong. They're strong enough to hold these bases together, but they're weak enough that we can come through with an enzyme and break those bonds without too much trouble. So that's a good thing. Running in opposite directions refers to the 5' to 3' direction of a DNA code, and I will show you that in a diagram in just a little bit. But know that our DNA strands do travel in opposite directions. And so here you have it. This is the DNA double helix. Here's thymine bonded to adenine and guanine bonded to cytosine. Here's the sugar group. Here's a phosphate, and it's bonded into the sugar group of the next base up the sequence. So this is called a DNA sequence right here. They're often called referred to as having a phosphate backbone because of these phosphates bonded to sugars that make up the frame of the ladder, if you will. And then it's twisted into this double helix structure right here. And then here, if you look real close, you can see 5', 3', 5', 3'. And so that's where you get these arrows, 5' to 3', 5' to 3'. So we'll look at that a little more later. But that's the structure of DNA. Very cool. So we're going to stop there. We've covered a lot of ground. We'll jump back into 8.4 next time. Thanks for your time, guys.